My name is Adi. I am from the Department of Biotechnology, IIT Madras. I'm here a faculty since 2012. I'm currently a professor here. I teach, do a lot of research, train students, and teach many of the undergrad students and master's students and PhD students. And that's what my primary role really entails. IIT Madras is one of the premier institutions in India, and it's actually ranked the first in many of the disciplines across the last 4 to 5 years or so. It's actually a highly sought after institute, and we have several departments within the institute, and biotech is one of them. And the biotech department has people working on different domains, and some of these domains are bioprocess engineering, molecular basis of diseases, computational biology, and even people working on protein engineering and protein dynamics. So there are many different streams. People work on diverse aspects and also across different time and length scales of the problems that include people working on tissues, live animals, to people working on molecular level details of proteins and DNA. And so we have a good mix of faculty, not only in terms of the domains, but also in terms of the age groups, if you want to say that. I do not know how to say this, but yeah. So the people working in different aspects and also kind of diverse in terms of their backgrounds and the times at which they have joined the institute. And we have many assistant professors, associate professors, and a few professors as well. So I believe we are on the right track, and I hope we can make a change in some way or the other. Our group is actually titled or called the Protein Biophysics Lab. So a lab deals with the biophysical aspects of proteins, and this would involve primarily protein folding mechanisms or the sequence of steps involved in the folding of protein. And for this, we do a lot of spectroscopy and spectrometry. One of the techniques that we use quite frequently in our lab, and it's almost like the initiation that any student goes through, is to actually handle a Chirascan instrument. And this would involve a lot of CD, circular dichroism measurements, fluorescence measurements. We also have top flow kinetics that comes with it, or an attachment that comes with it, and we use that quite frequently to measure both equilibrium properties and also the kinetic properties of protein systems. And the systems vary in size. They also have very different functional properties, all the way from antibiotic binding to protein binding to DNA binding. So we have a variety of systems that we work with, and of different sizes and shapes. And Chirascan has been quite helpful since 2015 or so, when we actually purchased this equipment. So it's on nearly 9 years that we have been using this equipment. Our work at IIT Madras at the biotech department primarily involves working with different kinds of proteins, as I said before. And these proteins are actually quite diverse in terms of their functionalities, and some of the aspects that we are working on require detailed characterization of not just their structure, but also their folding mechanism, and also the stabilities, and also the function. So we kind of try to understand how function influences stability, and folding mechanisms, and how folding mechanisms influence function. So we are trying to kind of go back and forth using a combination of techniques, and Chirascan is one of them. And many systems that we work on. One of my favorite systems is this protein called histone-like nucleotide structuring protein called HNS. And this particular protein is involved in condensing bacterial chromatin. This particular system not only exhibits differences in its structure as a function of temperature, but also exhibits varied oligomerization extents that depends on external osmolarity, temperature, and pH. So we are actually using Chirascan instrument, particularly CD and fluorescence, and the kinetic measurements that come with it, to actually characterize this in great detail. And this will give us some insights into organization of bacterial chromatin. And if you can understand this better, so you can actually control the extent to which bacterial genes are expressed or repressed. And we can kind of use even phosphoromimetic mutations and even peptides that mimic these phosphoromimetic mutations to actually control the expression of genes. And that's what we primarily do. This is one of the aspects that we work on. The second thing that I would like to also highlight is that we are interested in antibiotic sequestration mechanisms. It's one of the primary mechanisms that bacteria use to actually control the expression of genes, and also to control the fitness levels. So what we try to do to the extent possible is to understand the mechanisms or the steps involved in how proteins go about binding antibiotics and kind of sequester them such that the antibiotics are unable to exert their action. So the way we start off is actually purify a particular protein. We characterize the system, its spectral properties via circular dichroism and fluorescence, both in the presence and absence of the antibiotic. And this is one of the primary steps that we actually take to characterize this particular protein. And there are a few proteins that we work on. One is TPase, it's called the thiostreptin binding protein. The other one is albase, which is also called the albucidin binding protein. And we are working in these systems in great detail. One of the primary things that we like about Chirascan is the fact that, touchwood, it's kind of worked non-stop in many ways, and we have been using this, as I said, for nearly 9 years. I believe the data are incredibly reproducible across different time points that we have acquired for the same system, and so that kind of gives us some kind of confidence in the kind of data that we get. And there's a lot of reliability issues that comes with when you kind of switch between instruments, and with the same instrument, when you take the data or acquire data at different time points, there is always some issue or the other. But in this case, we have had no issues whatsoever. I believe every experimental work from our group has really exploited the system in one way or the other, where we have acquired fluorescence or circular dichroism data, including for near UV CD, fluorescence with the ligand, in the absence of the ligand, even anisotropy experiments and kinetics of DNA binding, and also even the kinetics of phase separation, which is something that people are interested in. The first time I learned about applied photophysics is from actually my PhD advisor, Dr. Munoz, who is currently in the University of California, Merced, and he was the one who recommended me this particular instrument, even the accessories that come with it. And he indicated that this could be a very good start because I just got this Wellcome Trust DBT intermediate fellowship, and he said I should be able to buy this particular instrument so that it kind of covers all the bases, including CD, fluorescence, and also kinetics, all of it which comes in a single setup. The customer support from Applied Photophysics and team here at India as well is quite excellent. We've been exchanging emails once in a while, and people have logged into a system and tried to fix it, and this happens many a time. But every single time we have been able to address the problem, and we have had no problems whatsoever in this particular. I would say if you're going for a Chirascan QCD, please go for it, and then and not hesitate. And simply because the instrument works very well, few hitches here and there once in a while, but it's almost the standard thing that happens with equipment. Otherwise, we haven't had any problems. The students have always found it quite easy to use the equipment, and it's pretty much intuitive. So we have had no problems whatsoever.